Welcome back to another episode of Bio to Bedside. Really excited about this one. Aaron, really excited to have you on and talk about the work that you guys are doing in the solid tumor space. I guess just before we get started, for those of you who have been able to join, we'd love to kind of learn where you're coming in from. So if you're able to sound off in the comments and just let us know where you're coming in from, that would be fantastic as we get started here. All right. So before Jeff has a little bit of a soapbox,
So much pressure.
We're going to, Aaron, would you be able to just quickly introduce yourself for everybody again and just quickly take us through what Kiragen does?
Yeah, absolutely. One, thanks again for having me after I randomly hopped on your last webinar. Always a pleasure to tell our story. So my name is Aaron Edwards. I'm the CEO and co-founder here at Kiragen Bio. And we're on a mission to use multiplex gene editing and AI-guided strategies to enable cell therapies to finally have durable efficacy in solid tumors, and stay tuned for liquid tumors broadly.
Nice.
Right on. Okay, so the plan is overall today to basically kind of go through what kind of makes solid tumors really unique and a big challenge, kind of the key weapons in the arsenal that we should be thinking about. Some people were liking the thumbnail I put out this morning, and the idea there being really you need to take a bunch of different weapons to really attack this big beast. It's not just one thing by itself that's going to work. And then finally, the taking those two things together, kind of the uniqueness of the challenge of the solid tumor and all the necessary tools and equipment and weapons we need, kind of what you guys are doing at Kiragen to kind of develop next generation therapeutics in this space. So it's going to be exciting. So looking forward to that. Folks, if you have any questions for Aaron on top of that, please put them in the chat and we will get to them as soon as we can. We want this to be a conversation with you, not just Ben and Jeff jammering on and talking to Aaron. We want to include you guys.
I love jammering on.
Yes, you do, Jeff. Okay, sweet. So, Jeff, in the context of solid tumors and everything that's going on in that space as it relates to CAR-T, there was a fairly big announcement, some big funding recently about a certain approach to solid tumors, and I would love to get your take on that. Fill us in, Jeff.
Yeah, so I don't want to take too much of the spotlight away, but I think that the conversation will need to start with an explanation of the challenges that CAR T cells face in solid tumors that they don't face in B cell malignancies and like liquid cancers in general.
So maybe just let's stop for a second. So for CAR T has so far found a lot of success in liquid tumors, like the blood cancers, right? Because the tumors are way smaller. It's not something you really have to deal with a really big, dense thing. You're able to just very quickly kind of disperse your CAR T cells throughout the body, find these different small little sections of the tumor of these tumors that are living in the bloodstream. Attacking them and eliminating is pretty straightforward. It's more difficult when you've got a golf ball sized tumor.
Kind of. So
more or less.
So things like lymphoma, well, I'll start by talking about, here's some of the things that most people don't really think about when it comes to like a cellular therapy. Like for CAR T cells, the cells, we think, okay, we're going to infuse these cells. They're going to go in and they're going to find the cancer cells and then they're just going to kill them, right? And then if they don't do that, it means that they can't kill the cells for whatever reason. There are a lot of things that have to happen before that, before you can even get to that point. Like, for example, you need to have the CAR T cells get to the site of the disease. Right. And being T cells, they tend to really like being in the lymphatic system and in the bloodstream. They don't really like to infiltrate deep into tissues. There are subsets of T cells that do that, but in the majority of T cells, they like to go to the lymph nodes and like hang out in various areas in the body, which means that when you have a cancer in those places, the T cells will kind of naturally traffic to those places. So when you infuse them, trying to get them to like a breast cancer tissue, is significantly harder than it is to get them to go to the lymph nodes and like find lymphoma cells, right? That's the first problem with solid tumors. The T cells just don't want to go there naturally. There is like chemokine signaling and things happening in the tumor that will kind of draw them in, like inflammatory signaling. But for the most part, getting to the tumor is like the first step. We got to get those things to the tumor. And then getting to the tumor isn't enough. You've got to penetrate and get inside. And with a lot of solid tumors, and this is the case for lymphomas as well, they can kind of form a microenvironment as well. But for solid tumors, especially, you can think things like glioblastoma or like lung cancer, pancreatic cancer. The vasculature, which is how the cells get to the tissue initially, is always messed up. Cancer will induce angiogenesis, making new blood vessels, but it's super chaotic and it's not like a nice like 6-lane highway. You can think of it as like dirt road, back roads. It's just difficult to get in. And then once they kind of get, and they can diffuse into the tissue, but then you can think you need to consider the fact that solid tumors have very high interstitial pressure. And they have dense matrices of stuff that the cells have to overcome all of these things to get deep into the tissue. And you got to consider the fact that the cancer cells don't want them to do this. Like if the cancer permitted T cells deep into the tissue, it wouldn't exist and you wouldn't know that it was there. So if a tumor exists, you can already be sure for the most part that tumor is pretty good at excluding things like T cells. Okay. That's the first part, is to get the things to the tumor and inside the tumor. It's tough. It's tough. Now, then you can start to have the conversation about like difficulty with antigens. And not many people seem to appreciate this, but the reason that hematological malignancies are such an easy target for CAR T cells is because what are the most common targets for CAR T cell with like B-cell lymphomas? It's CD19. Healthy cells express CD19 as well. It isn't a cancer antigen. It's a B-cell antigen. And you get B-cell aplasia. You just kill everything. And the issue, or I guess the good thing with CAR T cell in like B-cell lymphomas, is that you can survive with your B cells being depleted. It isn't like a lethal thing, right? So you can have the CAR T cell just kill all the B cells, the cancer cells and the healthy cells. How do you do that with brain cancer? Right? Yeah. Like if like tissue antigens even exist and you targeted like a liver tissue antigen, you're going to have your CAR T cells killing the liver. Like you can't do that, right? So you need to find like disparities in the expression of things, and it makes it super complicated to find the antigen. And that's why solid tumor antigens is so hard, because you need to find like a cancer specific antigen. And now this gets me to the conversation about Dispatch Bio. Dispatch Bio has a technology that they're calling Flare, right? And they're basically saying, hey, we want to make an antigen that is only expressed on the cancer cells. And then we can send in a CAR T cell therapy that will target and kill those cells, right? So then they make a virus that will preferentially infect and then replicate in cancer cells. And if that sounds familiar, that's because that's what oncolytic viruses do, okay? But then instead of having the virus kill the cells, because that wouldn't be a new thing, they have them express a protein called Flare. And Flare is apparently, it's a synthetic protein that is going to be embedded in the membrane, and the extracellular domain, they have a CAR receptor that can bind to that domain. So you can think of it as like CD19, but not CD19, right? It binds to this Flare protein, and then that's the target. And the idea is that we can have the cancer cells express Flare, nothing else will express Flare, and then the T cells will go in and kill the Flare positive cells. It sounds good if you think about it for like 1 second. Like I think you think about it for 1 second and it's like, yeah, this makes sense. Like, what's the problem? The problem is on top of the things that I just talked about, where like half of the problem is getting the cells to the tumor in the first place, right? To find these cells. If we have achieved viral mediated cancer specific expression, why are we using that opportunity to express a non-lethal surface antigen when you can just express some kind of like apoptotic induction? Or, you know, the rebuttal to like the apoptosis induction is like cancer cells tend to have very like anti-apoptotic signaling happening. So it would need a very high threshold to induce like caspase activity. Okay, then pick something else. Like there are like tumor suppressors. There are CDK inhibitors. There's all kinds of like really potent things that will stop cell division that you can express in these cells. And then if the answer becomes, well, we don't really have super tight expression in only the cancer cells, so it would cause toxicities if we expressed like, I don't know, some kind of like a P21 in these cells, right? My rebuttal to that would be like, if you don't have cancer-specific expression of your cassette, you're going to get flare expression on the healthy tissue. And then what's the difference between, you know, what have you actually solved? So now we're saying, okay, they're going to express flare when they could be just expressing something to kill the cells, which would be an oncolytic virus, right? They also claim that flare expression is going to combat immunosuppression. I don't really know how. The only thing that I can think of is one of two things. One, they're not saying everything. They're leaving something out. Like maybe they're also going to express IL-2. I don't know how they get this immunosuppression combating, whatever. But what might be happening is flare itself is a synthetic protein. It's not found in nature. So like by having your cells express flare, you may be able to induce an immune response against that foreign target, right? Okay. But the problem is that to get the CAR T cell therapy, what do you do before CAR T cell infusion? Lymphodepletion. So you're depleting the immune system that you're then going to be relying on to have a flare-mediated like immune response. The only cells really that are going to be available are going to be your CAR T cells, which only kill flare-positive cells. And so hopefully I'm kind of conveying, I'm not doing it very eloquently, but like hopefully I'm conveying the fact that this is not the universal solid tumor CAR T cell therapy to cure all cancer. It's like, I actually don't, I used the analogy with my wife earlier. I said, imagine we have a situation where I'm building a door, and I'm hammering in nails, and my hammer breaks, right? And I say, oh, I need a way to hammer in my nails. And somebody comes along and they say, here's this door to use to hammer in your nails. And I say, well, I don't need to. I have the door now. Thanks. Mm-hmm. Right? Like I wouldn't use so if the goal is to kill the cancer, if the goal is to have CAR T cells work in solid tumors, this is, I guess, a method to do that. But the goal isn't to get CAR T cells to work in solid tumors. The goal is to kill the cancer, right? And if we can get cancer cell specific expression, highly expressed flare protein, just take away the flare, take away the CAR T cell therapy, and just do an oncolytic virus. What's the difference?
So if I can jump in here. So again, I guess the main thing we want to emphasize here, right, is just when it comes to solid tumors, it's just such a hairy, difficult problem that if there's probably just not going to be one solution that can just fix everything, right? And so that's kind of what we do want people to take away from this. Some pretty, like, it's somewhat interesting to see what's happening with this flare protein, but the challenge is going to be, is this really going to be something that ends up being valuable and useful in that overall plan of tackling a solid tumor effectively and ultimately getting it out of and getting it eliminated in a patient? Is that fair to say?
And I will say this. Now, this is a good segue into Kiragen, because in order to get solid tumors to work, or to get CAR T cells to work in solid tumors, we have to overcome, like, let's say three things. We'll say three things. First, we got to get the things into the tumor. Okay. Then we have to overcome the immunosuppression. And then we can think about antigens. Okay. If you don't do the first two things, nothing matters. Nothing's going to matter, right? So with Kiragen, it's my understanding that you are directly, with your infusion method and with your multigenic approach, we are now kind of entering into a conversation where we can start to take this stuff kind of seriously, where we understand the complexity of the microenvironment. We understand that it's going to take a multifaceted approach. And one of those things is finding good antigens, but there are several layers to this that's going to require complex things and complex strategies to overcome.
Yeah. So maybe before we hit the Kiragen thing, let me comment briefly. As someone who is currently trying to close a funding round myself, it is great to see an investment in the solid tumor CAR T space.
Right.
Period. The Parker Institute is known to be bold. They say in their press release they do bold things. They're funding bold science, and they are clearly aware of the questions we all have. I think where the discrepancy comes from is unfortunately how some media portray this as the silver bullet. It's great PR, but I think just people need to be cautious that there are lots of challenges to be overcome. And I'm not an oncolytic virus expert. I don't have anything more novel to add here, but we will see. It's a lot of money to get into the clinic, but it's complex therapy or dual therapy in one. So stay tuned. That's all I can really say.
So I do agree that seeing life in this space is amazing, right? I did the exact same thing with in vivo CAR therapy. I'm trying to be like the voice of reason, I guess. I hate to like position myself like that, but there are a lot of people that I see where it's like we found the answer. Like this is insane. Oh my gosh.
Yeah.
And it's like, oh no.
And I think this is also the challenge. People are almost forced into messaging that they found a silver bullet. Like saying you cure cancer is not appropriate in the science field. And when people ask us about in vivo CAR T, for glioblastoma, in vivo CAR T is not ready to be deployed. And that's why we're not doing an in vivo approach. Even if they could deliver the payloads, we're doing ex vivo. So it really needs to be this conversation around all these tools. And Carl June has a really great article that he talks about this, actually, where there are best fit tools for each indication, because every cancer has a little bit something different that might be the challenge, which is why we built the platform the way we did.
Sure. And I want to address, I've seen this comment come up a few times now, this idea of immunogenic cell death. Okay. The CAR T cell mediated killing is not the only way to induce immunogenic cell death. Oncolytic viruses also induce immunogenic cell death. Yeah. Chemotherapy can induce immunogenic cell death. All immunogenic cell death means is that when the cancer cells die, that death can result in the release of DAMPs. And DAMPs will activate the innate immune response and the adaptive immune response. But really, it's the innate immune response. And immunogenic cell death can be induced by all kinds of different things. So this idea that we're going to be getting CAR T cell induced cell killing, which is a way to get immunogenic cell death, that's wrong for two reasons. One, again, it isn't the only way to do it. And number two, you need to undergo lymphodepletion prior to CAR T cell infusion. So any kind of immune response you're going to get is going to be naturally hindered because of your depletion of the immune system. So oncolytic viruses already do that explicitly, and there's no requirement for lymphodepletion. It's like part of the cake with oncolytic viruses is this immunogenic cell death. So I hear, I get that we're kind of like grasping for straws now, but there is some problems with the analysis that I've been seeing.
Yeah, I would say finally on that cell death comment that just like in vivo approaches, if it hasn't completely resolved the tumor, you're still going to get your immune cells being suppressed by the very things that we also protect from. So at the end of the day, I think it's a bold leap, and the only way to know what will happen is if they put the experts in place to figure out all the pharmacokinetics of what they would need to have happen to make some sort of therapeutic success. So stay tuned.
Very good.
Yeah.
Okay. So we've accumulated a bunch of comments. Want to take the time now to kind of address them in a batch, and then what I'd love to move on to after that, right, and to really get into it with you, Aaron, would be what are the different tools that we should be looking at to make CAR T effective in solid tumors. So just before we get to that, let's quickly go through some of the questions here. So first of all, Stuart, I love this. I know I put this comment up already. Got CD19 t-shirts for those that remember that style. Ryan Murray was saying we should actually put CD19 isn't an actual cancer antigen on a t-shirt. Jeff, we should do
or
that.
BCMA or CD20 or any HER2. Like none of these are cancer antigens.
Right on. Okay. And then So I guess the interesting thing here too, right, would be with that flare protein, like you'd have relapse after flare being high because it's not something that's consistently expressed. And so would there kind of be like an immune evasion play here that you might have to consider?
If anything, the flare expression will lead to, like when I say flare expression will lead to a flare targeted immune response. And I don't mean by the CAR T cells. I mean by like our natural adaptive immune response, because flare isn't a protein that we make in our bodies. So we wouldn't have tolerance for it. The only question becomes, are there like MHC compatible epitopes in the flare protein, so where if it is processed, can it be loaded on MHC and like induce a response? That maybe, if you just like took a virus and expressed like some random amino acid chain, it would be foreign and it may induce an immune response, but it's not guaranteed. This the idea that once flare goes down, like, because it's not a permanent, I'm pretty sure it's AAV, so it wouldn't be like a permanent expression. But yeah, if that's what he means, it wouldn't cause like relapse if flare was high or low.
Okay.
Yeah.
So maybe to follow on from that, there's an interesting comment here from Shireen. Yes, dispatch is also expressing cytokines to the oncolytic virus. I believe that was in one of the press releases. So does this would this help a little bit just in terms of diversifying your
So there's two problems.
memory?
There's two problems. It isn't oncolytic. They are explicit about that. It's not oncolytic, and in order for, because like what'll happen is the cells get infected and the virus will replicate in the cells, but somehow it's not oncolytic, so it's not going to like burst the cells. They show that the only way to release viral particles is through CAR T mediated death. So it isn't an oncolytic virus. The idea of having they will also encode like an IL-2 or whatever or TNF alpha onto the viral genome. That's what oncolytic viruses do. Like, because then the cancer cells themselves will be expressing the IL-2.
I think one of their, correct me if I'm wrong, whoever said this, but I think their cytokines were chemokines. If I read one of the press releases right, but don't quote me on this. I think that's to help for
it's with the infiltration. Yeah.
Okay.
Yeah, which that, if that is what they're claiming to overcome immunosuppression, right? Like we express CCL21, right? That will help with trafficking, but it's not going to overcome immunosuppression. It's just the point of this whole diatribe that I've been going on is that this is not the answer you'd be looking for if it was like some kind of universal CAR T cell therapy for solid tumors. It's just, it's so complicated. There's so many layers to the immunosuppression, and like the fact that there's so many layers to the onion of like CAR T cells don't work in solid tumors. It isn't just like, we got to find a better antigen. That's not the problem. There's like 4 or 5 like big problems. And when you see things like that, I would expect to see like a bunch of things happening. And it's like, we have flare. It's like, okay, what else you got? So.
Okay. And then one final one for right now. I put this one up on screen before, but I just wanted to get to it since it's a bit of a earlier one. This chain reaction theory is problematic in many ways. And you're right. Why not using the technology to express something more efficient than an ag?
An antigen. Yeah.
An antigen.
I think it was more of like a Jurassic Park situation where the scientists were so concerned with whether or not they could do it, they didn't consider whether or not they should do it. It's like we're trying to get CAR T cells to work in solid tumors. That's the goal. That's like their goal. But the goal isn't to do that. The goal is to kill cancer.
Yeah. All right. Perfect. So again, Aaron, we want to make sure we're making the most use of your time here too. So if we just move on quickly, thank you everyone else who's put in comments and questions so far. Our plan is to get to them. We just want to make sure that in the time that we have, we're able to kind of get through some of the key components of the show today, and then leave a bit more time at the end for Q&As. So please keep them coming. We're planning on having some time reserved at the end for those. But yeah, Aaron, if we're just kind of doing a bit of a plain language summary here for the different tools that we should be thinking about for attacking a solid tumor with a CAR T, what would be these different key bins or baskets that people should be considering? What are the different weapon systems that we should be using to tackle this problem?
Yeah, Jeff hit the nail on the head here, and how we describe it to date. There are 3 buckets that I describe that are the largest challenges to why solid tumor cell therapies have not worked. It's getting the cell there. How does it get there? What does it target? The story is a bit different. I have a lot of comments on CD19, regardless. And when it gets there, what do you do about all those things that can shut off cell therapies? Like, those are the 3 buckets. I would say a lot of progress has been made in the first 2. So, or there are indications like glioblastoma and gliomas where they skip a lot of that and have actually found a way to have local delivery. So, it is indisputable now, not maybe 5 or 10 years ago. People really questioned if these T cells even got to the tumor site. So for many reasons, every tumor is different, whether those physical barriers exist or not. The reason that sparse T cells get to these spaces is because, again, just what Jeff said, they're not attracted there, or fewer are attracted there because some on the way are stopped and turned off. It's pretty clear they can get to the solid tumor and they're turned off. So what do you do? I mean, almost instantly they're turned off because these tumors have glioblastoma is one good example. They have 50 to 80 different pathways across multiple buckets of just the tumor microenvironment types of activities where they've overexpressed or underexpressed things that evade your immune system and these immune cell-based therapies that we can make externally or internally now. So, I'll ignore a little bit on the first 2. So people have made progress by doing more than one target of the CAR. It's pretty clear, even in this liquid malignancy space, you should be targeting more than one thing at a time, because that prevents or at least increases the chance the CAR T can continue killing the cancer cell before it can remove those proteins in some different way. CD19 is a great example of that. The trafficking piece, people are working on those things. They have cytokine or chemokine overexpressions, or like GBM and others, you can do a little bit local regional delivery. The third piece is a little bit unfortunate that it's one bucket. The tumor microenvironment is this holy grail because it's everything. It is innate immune cells that come in that are maybe not normally there, that turn off that activity. It is your checkpoint inhibitors, your PD-1s, Keytruda. Think about those pathways that are blocked. All of those different pathways are upregulated. It can shoot out suppressive cytokines like TGF-beta, adenosine, really bad things that just make this environment toxic. And for immune cells, except for cancer, they thrive in that environment, and low oxygen. So when these cells, even if they penetrate and start doing something, they don't do it for very long because they don't have nutrients, they don't have oxygen. It is just a really hard problem. And that's why we focused all of our approach for now on that third bucket of just the tumor microenvironment and how we can protect against those factors, because a lot of people have made progress to date on the first 2.
Excellent.
And so, if I can give a little bit of a story to help kind of give people an idea of what the microenvironment looks like. A lot of people, this goes back to like the 80s. A lot of people think of tumors as unhealing wounds. And when we get a big wound in our body, our immune system is drawn there immediately. Things like neutrophils and macrophages and like various myeloid cells are brought there immediately to clear out any kind of bacteria that may have gotten in and to initiate infiltration of fibroblasts and things like that, which will start to create all kinds of like matrices. And you can begin to think of like what your body does when it heals a wound, right? It wants to get rid of the bacteria, build new tissue, and then suppress the immune system. You don't want to have inflammation there all of the time. So it comes in, starts it really fast, and then it shuts it down. That's what a tumor is stuck in that spot. A tumor is stuck in the dense fibrous matrix in the immunosuppressive part of wound healing. And it's being mediated by cells like macrophages and neutrophils. And they're not doing it because they think, oh, well, cancer is here and we're going to help it. No, cancer hijacks the wound healing process. And you get a lot of, you can just think of it as like an unhealed wound, and it's a big mess.
Yeah, and I think it's also it is complicated. So maybe one correction from the intro: I don't think we fully understand the tumor microenvironment, but we know enough about it to start addressing some of these things more comprehensively. That's what maybe I'll say. A really good example of what you're talking about in the wound healing: glioblastoma tumors are like 40% by weight macrophage myeloid compartment.
Yep.
So like when people think of a tumor, it's not one cell. That's also what makes it really complicated to target and protect from. But there are overlapping things that not only the myeloid compartment, but the tumor driven suppression itself, there are overlapping things there. So if you're protecting against one thing, you actually are sort of already creating multifaceted protection from sort of the different pathways or different cell types that might be inhibiting your cell therapy or immune system.
Okay,
yep.
Yeah, so one additional question here then, Aaron. So if we're focusing in here on the final step 3 of that bucket, the tumor microenvironment. What would you say are the main buckets there for inside of that, in terms of where we should be developing weapons and systems to properly address this issue? Just to kind of do a little bit of a higher.
To date, there are a couple buckets. So a lot in the synthetic biology department. So they would say, let's take one of these pathways. So instead of blocking it, they might co-opt that pathway and say TGF beta. We're going to put a receptor next to the CAR that sees TGF beta, and every time it sees it, it does something to the T cell or not. So it can either create what's called a sink, so it just like sucks up all the TGF beta, or sometimes when it binds TGF beta, they'll have a signaling component that is in addition to the CAR. So a lot of these different types of strategies, you'll hear about flip receptors,
switches,
the switches, all these different types of synthetic biology approaches that could be targeted to one or multiple of these pathways. That is one approach. I think we think a little bit differently around CARs are not natural molecules. They're not natural. You're co-opting some T cell signaling, but we try to avoid changing the engineering of the T cell even more than what it was naturally designed to do. So when you start overexpressing cytokines or putting more signaling components in T cell, there's just a lot of uncertainty about how that's actually going to behave in certain environments. So synthetic biology is one of them. The other big bucket would be gene editing. So we are a multiplex gene editing company, and within gene editing, there are also different strategies. Do you knockouts are the primary sort of approach here so far, or some that are coming into the CRISPR activation space, or epigenetic space. For knockouts, you can either do something internal to the cell. So to date, a lot of the companies that you will see that have products that are into the clinic now, they thought, or they're sort of on this dispatch bio train of having sort of a silver bullet approach. So minimize the modifications you make. And if you minimize the modifications you make because you shouldn't make more, if you're cutting, you shouldn't make more. Or if you just don't like the theory of making many modifications, you change the potency of that cell. So you want to say, take this T cell, and when it gets to that tumor, you want to make it as strong as possible to just break through. You do this by removing some regulatory breaks. So there are actual breaks inside the cell that naturally attenuate that immune response. So you can, and there are many of them, but you can knock out one of them. And so every time the CAR signals, it signals more than it normally does. Or what we do is just knock out receptors on the cell surface that are seeing these different suppressive pathways or factors. And if you know how to combine these knockouts together, instead of having a silver bullet approach that fits all, you can say maybe there are signatures, but in general, you can sort of tailor each of these knockout combinations for where they are going, which is the brain or the liver, where pancreas, wherever they might be, and just prevent suppression without actually changing the potency of the cell. I'm not going to try to pitch every time I say this, but if our goal is to take the efficacy that we've seen in blood-based malignancies into the solid tumor space, not to change, we don't think you have to change the CAR any more than it already is because it works really well, But if you enable durable functionality longer than it can today, you can possibly enable actual sort of, say, I would hope, curative outcomes, but really move the needle here for these patients without actually supercharging in that direction. So two big buckets in bio and gene editing.
So one of the things that that I would immediately think of is we are not sure how a highly robust large population of immunostimulated T lymphocytes that lack TGF beta receptor and lack PD-1 and lack other kinds of breaks will respond to activation in the body. They could transform, they could become highly toxic, because the immunosuppressive mechanisms are put in place on purpose by our body. So to remove all of them, with the one goal of saying we're just gonna like get activation and like really just like drinking from a fire hose, get these things to become activated, is there potential to accidentally actually cause more of an issue with too high of activity?
Yeah, it's definitely a concern. They also fall in two different buckets. So our approach of protecting against suppression is meant to not overactivate, because you can't overactivate even more so in the brain, where there's already a lot of concerns from those signaling components or just immune activation in general. Those receptors, as you said, are equally seen as these sort of immune breaks, but they're a little bit different. So they dampen the immune response, while these internal breaks, if you don't have them, they will actually cause sort of supercharging. So they can cause a little bit different activities. I would say the tool, if we're sort of circling back to tools, regulators in some other programs have required safety switches. So if you start seeing some of that high activity, there are ways to turn them off. That is a challenge, because when you start turning these off, then the CAR T stops working. So it really, what we see this as too is just like the early days of liquid malignancies, it is just as important for the tool to be matched with a best-in-class clinical team that knows how to manage these therapies, because it is a clinical management puzzle to be solved in a lot of ways. But hopefully we're providing those teams with the tools that put them in the best positions to succeed.
That's a good point. I wouldn't have thought of that, and especially with like, so do your products include like a kill switch?
They don't currently, but I would admit that we haven't had a pre-IND meeting with the FDA, so it's something we know that they've asked other companies to do. But specifically, they've asked other companies because they supercharge. So if they supercharge, you have to have something turned off. Admittedly, we are kind of front loading our strategy before we even have these meetings, knowing that we will have a lower dose to start in our clinical trial and then work up, because that is one way to solve this. I would also say, I think I would say stay tuned. I shouldn't say too much about if we will or will not have a safety switch in there, but it's something that is top of mind.
Yeah, I'm not super keen on. Sorry, one more thing, Ben. I'm not super keen on these kill switch ideas, especially because I feel like the real chance of transformation and like CAR T cell mediated like malignancy is like insertional mutagenesis of the CAR, which is so rare that it isn't worth like, oh, everyone needs to have a kill switch. It's like, it's not really what you think it is.
I should have mentioned that too. That's a really good point. The other reason why we're really excited about what we do is that the concern in some of these other with these other companies, they're not only doing this complex engineering, they're doing it in patient-derived cells, which have a higher risk of this insertional mutagenesis. So usually all these cases in the news where something has happened, if you really read under the hood, these patients had a mutation already that was really, or it was detected just not at high enough levels. And if you knocked out the other part of the gene, then that's what is causing this mutagenesis. We are using healthy donor-derived material, which have a significantly, I don't even know how to describe, less chance.
Infinitesimally small.
Infinitesimally smaller chance. And so when you put these cells in, the sort of knockouts and the combination of knockouts, we are not concerned that that is going to have the same type of risk as what people have seen in very, very, very rare circumstances of these secondary malignancies.
Okay. So so when we're thinking again, just kind of about the different weapons in this approach, then what we're looking at then mostly is just how can we prevent the solid tumor from being able to negatively influence the CAR Ts from doing what we have a plan for them to do. And the other component here being as well that given that each cancer is a little bit different, you guys kind of have a mosaic of these different deletions, these different things you can kind of like these abilities to immunize yourself from peer influence for a specific cancer so that the CAR T therapy is going to be much more effective for a specific indication.
Yeah, absolutely. And I think, something that is rarely talked about is, I think it's not trivial. T-cell biology is very well understood at the sort of mechanistic level. What we've done in the biologic space with Keytruda, all these other products is really great. The pathways are well known, but the question is not discovering I shouldn't say flare protein, but not discovering something new that people haven't done before. It's how do you already take the information that we know about T-cell biology and combine that in a way that makes these cells best suited for this tumor microenvironment. So for us, it is, let's just say PD-1 in the checkpoint inhibitor pathway, let's say TGF-beta or, those combinations. How can you target and protect against non-overlapping suppressive pathways? That's a cool concept, but it's something that is done in singlet, say one of those separately. Plenty of people have shown, you knock out this, it makes this CAR T better. Okay. Actually, when you start combining them, they don't all talk very well together. That's really important for people to know. So when we talk about what we do, it is not just the biological ability for us to multiplex edit safely and effectively, which is awesome. That's great know-how for now. It is the rational decision-making of what we pick and making sure that is also best suited for these spaces. And I'll finally, so I don't go on a diatribe here, the tumor microenvironment is dynamic. It is living. It is adapting all the time, not only constantly over time, but to the treatments that you provided. So one thing that you see all the time in LinkedIn and all these papers is that for a long time, I just say Keytruda and PD-1 is a really great example of this. Just because someone had a biopsy of their tumor, they saw PD-1 or PD-L1, does not mean that they will respond to Keytruda. And people have wondered why is that? There are a lot of reasons for it that we don't have time to go into, but one of them could be this dynamic tumor microenvironment. So not only are there like soluble PD-1s that can be blocking this, but if you are PD-1 negative here, you get treatment, maybe you're PD-1 positive after the treatment. And so it is dynamic and changing. So our whole thesis is that you can't just attack one pathway or block one pathway. You have to do more. The biologic space is very clear about that. They wish they can do a Keytruda plus a CTLA-4 plus a VEGF inhibitor plus all these things you're seeing, but you get a lot of toxicities using biologics to block these pathways. So we've co-opted those pathways or the knowledge they have and just protected ourselves against them so that there's not this toxicity because it's just protecting against that suppression. And we do it at a scale that if the TME is changing, hopefully we've also enabled the cells to be continually protected against what the tumor microenvironment might be able to shift away from.
So that sounds complicated to get the biology right. How do you guys do that?
Yeah. Let me take a sip before I go into it. So it is a two-pronged approach. One, we are really fortunate to be building on top of clinical efficacy signals. If we did not have groups like University of Pennsylvania and Donald Warwick's team, Steve Bagley and MGH team and people at City of Hope that have taken these products into the clinic with single antigens or dual antigens and learned from those experiences, we wouldn't have anywhere to start. So it's one thing to look at a textbook and pick 4 pathways to go from. We can actually learn from their clinical experiences and know that at least the sort of final selections are validated to be really good targets to go after for glioblastoma. That's a starting point. It's empirically impossible to actually go through all the permutations that might be in that list. And that's why we're also building a machine learning guided platform to steer us in those directions. So what do I mean by that? We are not drug discovery for machine learning or AI, and we're not sort of on the clinical patient-derived side. We are at design optimization. If I told you that our lead product is 6 edits and there are 50 to 80 different pathways that you could pick, that is over 10 billion or something permutations that you would have to test to get conviction upon your best combination. That is debilitating and paralyzing for drug developers. So we are building this platform to steer us in the direction where even if they're bad combinations, it can tell us which combinations would be best to test in the wet lab to then feed back the machine learning model to have better predictions. And so it actually tells us, oh, you should probably test these. These are probably good. These are bad ones, but it's really important to know the bad. Not just from an IP perspective, but also just to be able to feed the model and say, oh, these are actually bad combinations. And again, that might sound really complex. To distill a little bit more, I'll get the question, okay, is this patient specific? Is it indication specific? Are there any that are we going to have a universal combination that's going to work everywhere? So I'll already get ahead of that. So the answer is it's not patient specific. Our goal for our glioblastoma product is to have a combination we know is as best fit as possible. But that means that you also don't have to say have inclusion criteria to confirm every single pathway is being blocked. You can say it is highly likely based on all of our data that this will definitely be beneficial for these patients. The cool thing though is if we go beyond glioblastoma, there's a lot of work on tumor microenvironment signatures or archetypes, I'll say. Mm-hmm. So if you bear with me for 20 seconds, I'll pause. But the dream with these off-the-shelf therapies is to say, if there are 12 archetypes of tumor microenvironments, we can have a dual CAR T, so to speak, that has tumor archetype A all the way through whatever K or whatever 12 is. And that can be best fit to the patient. So, it really is, and that might actually go across multiple tumor types because these CARs are pretty well validated, actually. Like there are tumor targets for solid tumors that are really good that have almost no expression in healthy tissues. But the way to get those to function has been elusive to date. So we are using validated targets in terms of the CAR and just enabling those CAR T to hopefully have efficacy so you can start seeing something to build upon in the clinic.
So maybe it will make more sense if we start talking the actual specifics. Can you talk about like what is like the lead candidate for glioblastoma? What are the edits? What is the CAR target? Can you kind of like talk about that or no?
Yep. So we're doing the exact same antigens that were used in the University of Pennsylvania trial, which is EGFR. But it's important. It's not just the cancerous antigen V3 that a lot of people talk about. It is an epitope that is shared across not just V3, but a cancerous conformation of EGFR. So it sort of has a dual purpose. So if you really dive, underneath, the reason MGH also has a dual approach is that there are cancerous sort of wild type versions of EGFR, and you need to be able to target that. So that's one target or one binder that can see both, only cancerous epitope. And then IL-13 receptor alpha-2. Mm-hmm. So one of these has been pioneered by Christine Brown, a lot from City Hope, EGFR V3 between Don O'Rourke and teams at MGH. They've really led the field with that target. Together, that solves, or at least checks the box of tumor heterogeneity. So our CAR can target and continue targeting not only more cancer cells, but if it expresses both, has a better chance of actually killing that cell. For our editing combination, since they're off the shelf, we are removing TCR alpha or TCR, so endogenous TCR, so that the product cannot have graft versus host disease, which has been very clearly seen. That edit is enough to prevent that.
Yep.
We actually have, it's really cool with TCR. Little known fact about this, it serves a dual purpose for us. So not only does it create these off-the-shelf therapies, but this myeloid compartment that's really, really significant part of glioblastoma and some other solid tumor types they exhaust T cells primarily through the TCR. So if you connect some dots here, really cool. People ask why allo or why these off-the-shelf therapies. Not only is it easy for us to knock this out, it also prevents this primary path of exhaustion that is going to protect our cells from like the last frontier of suppression in these tumors. So
I want to circle back to this TCR mediated exhaustion.
Yeah.
Okay. I won't go into it right now, but I want to circle back to that.
Yeah, well, happy to. The second edit that we have dual function, we'll say, is FAS. So there's been some papers that actually came out this week, but there have been several in the field that have been trying to do a knockout that might be similar to other approaches that try to knock out, your MHC1 and 2. And if you do MHC1 and 2, then you have to overexpress some sort of NK inhibitor. FAS actually solves all of that. It can actually, it's equipped, and that's what I was mentioning in the last call or hinting at. FAS is a tumor-driven suppression. So sorry, I misspoke. This paper that came out this week tried to say that FAS is actually a self-regulating. So FAS ligand and FAS upregulate on these T cells and CAR T, and they like cause cell fratricide. But and so for many solid tumors, FAS is not upregulated. In GBM, it actually is. So it is a suppressive pathway, and it gives our cells enhanced allo functionality that is similar to the really complicated engineering strategies that other companies are doing by knocking in and knocking out something.
So let me make sure I got this straight. What you're saying is that the cells will simultaneously overexpress FAS and FAS ligand, which causes fratricide.
Yeah. So this is another regulatory break.
Yeah.
So when natural T cells, endogenous T cells start signaling, there are so many different pathways that are supposed to dampen that and control it. It's a good thing to have these in place for the most part. So when we knock out one or knock out 5, there are 50 others that can also compensate. That's why we are fine with our strategy. FAS ligand, specifically in this paper that was published, I think yesterday or this week at least, when the CAR T starts signaling, it upregulates it even more. So then they are making a really strong claim that CAR T are actually inhibiting themselves. And so if you block that, then you are actually enhancing activity by durable enhancement, not sort of per cell.
Sure. Yeah. And for those that don't know, FAS and FAS ligand are like an apoptosis induction thing, where I'm not super sure if it's like the immune cells express FAS or FAS ligand, but it's a mechanism for immune cells to induce cell death onto cells, where you can actually have an interaction between FAS and FAS ligand. And if the T cells simultaneously start to express FAS and FAS ligand, that means that if I express FAS, I can see FAS ligand on like my comrade, some other CAR T cell, and just kill that CAR T cell via FAS ligand. So you guys delete FAS.
Yeah,
which disallows that whole process.
TCR and FAS, so it's making them off the shelf. It's making them a little bit better. So it's a still knockout only strategy that will be similar to other components in the clinic. And then we have 4 additional edits that are purely TME suppression. So PD1, common, LAG3. PD1 and LAG3 work very well together. PD1 and some others do not, I should say. TGF beta is probably the most, TGF beta receptor is the most common you'll probably see in the tumor microenvironment people work on. We have that blocked. CRISPR Therapeutics has that in their product. A ton of people, because that is a really suppressive, universally suppressive almost, I would say, suppressive pathway. And adenosine. Adenosine is rarely seen out there, but it's extremely suppressive.
Oh yeah.
And it's hard to measure, so that's really challenging.
I like your strategy of we do not, because I hear a lot of these things like the TGF beta, adenosine. A lot of people have said, hey, we got a lot of TGF beta in the tumor microenvironment. Let's make a TGF beta receptor switch signal that will basically act like a CAR, but instead of binding to an antigen, it binds a TGF beta. And what you're saying is that we're better off just leaving the T cell to do its work naturally and removing the brakes. and
Yeah,
not putting in any kind of like synthetic activation.
And the reason that I say this too, there is actually a paper in the heme space. It wasn't shown in solid tumors. I'm sure it'll come right after this. A group actually showed that if you just focus on TGF beta, whether it's a sink or it's sort of these overactivating signaling components, the tumor microenvironment shifts to become more suppressive. And so it actually like senses that you're removing TGF beta and then shifts to something else.
That's interesting.
And so, I mean, that it really proves the point. We have data in-house. You have to do multiple of these non-overlapping pathways, or it just goes to the other one and compensates for it.
See, now, I'm not sure how philosophical I want to get, but that to me seems counterintuitive, right? And it's important. I'm in love with science for this reason. Science was birthed out of the seeking of like what nature is actually doing and not just like what we think it is, right? So like ancient philosophers would think about, here's what we think nature should be. It's why we have circular orbits of the planets from way back when. And science was birthed to say, we don't really care what you think nature is doing. We want to know what nature's actually doing. And hence like experimental observational science was born. And what you're saying is that our observations are showing this thing that it seems kind of counterintuitive with TGF beta that you wouldn't think to kind of account for if you hadn't done this analysis, or at least ran it through whatever your process is to find this.
It's not dissimilar in some ways to almost like bacterial adaptation to antibiotics, right? In that if you're only ever targeting one specific thing, you're going to apply pressure towards a population that's not going to be as vulnerable to that one specific thing. And so what makes it far more effective is to have that combination approach to make sure it doesn't have different routes that it can immediately turn to, right?
And rational data mediated.
Yeah, and this is, I should do one quick shout out. I know a publication's coming, but she's presented on this, so I can say it. Investigator Dana Silverbush from University of Pennsylvania worked really closely with Don O'Rourke in that dual CAR-T trial and has some amazing kinetics showing over time at pre and post CAR-T infusion, all these different cell types and their compositions coming in and then rebalancing over time. And it's just showing this dynamic nature. It is really dynamic coming in, the CAR-T are cleared, and then all these other cells kind of leave. It just it is really challenging to understand this and adapt to it. And the final call out is this rationalization. It's really important for us to have some way to rationalize this. We can have a whole talk about it, but having some models that give you any predictive capability before you go into the clinic has been really elusive. And so our machine learning model actually can be trained on these types of assays that have phenotypic-based outcomes, including cell-based assays or organoid-based assays. So that Don's team and I should just say, so many people involved, used patient-derived organoids. So it's been a thing that's come up a lot. Organoids that actually more represent these different cell types and all of the tumor microenvironment components, put the CAR-T on them, ran their assays, ran the trial, and have actually published some clinical correlates to show that these organoid-based assays could be avatars for some sort of
That's excellent. Activity. That's excellent. And I wonder
how
our
model happens to train on them.
I wonder how. Sorry.
Yeah. No. Go ahead.
So I wonder. I've been on like the mouse models suck soapbox for a long time. Or patient-derived organoids is like the way to go. The only question becomes how quickly can you actually establish them patient by patient? And is it feasible but that goes back to the actual archetypes, I think, of your microenvironment archetypes we can make some generalized claims.
Yeah. So I would say, his paper would show, that you could sort of have this process that would be patient derived to then predict, should we actually give this patient this CAR-T will it work? Our goal is to actually have many different patients and learn from them so that we can a little bit, I would say generalize is a strong term, but then say we have a really good chance with this edit combination to actually treat all of them. And that's our goal, is that it will learn over time. And actually the pin for these transformer-based models is that it learns not only this, but if you start giving it all the data from other indications, then you can start asking across diseases.
Indications.
So if you really see the vision here, it's not just across diseases. One of the axes on the machine learning model, invite me back for more, is that it can actually start integrating cell type. So maybe not alpha beta CAR T cells, maybe it's NK cells. And the dream is to then have this as actually the hub of our entire company going forward.
That's excellent. That's excellent. Yeah, that's super interesting.
That'll be the teaser.
Yeah, I guess.
No, I guess quick question for you, Aaron. Do you have a hard stop or do you have a couple extra minutes? No worries.
I have a couple extra minutes, yeah.
Okay, excellent. If we stop kind of the conversation there, we've had a lot of people with some really fantastic questions, and so it'd be great to kind of get through those before we wrap up our time together. So appreciate everybody who's able to stay a little bit past the hour, and thank you for your patience. Let's go back through some of these different questions. Okay, so first of all, if we run back to back up here for J. Robert Chang. Thank you for questioning. Well, it's not a question, but a comment, and I'd love to get your thoughts on this, Aaron. One of the issues with solid tumors, their heterogeneity. Mechanism for transfecting one tumor cell usually doesn't translate to all tumor cells in the tumor, right? And so this kind of comes back to the dispatch. Yeah,
dispatch. Yeah.
Well, I mean, and also just even in the context of what you've just been talking about too, Aaron, right? The ability for you to now start to look at a multiple approach where you're looking at many different edits that are going to assess some of that heterogeneity is where we really start to see some interesting stuff happen.
Yeah, and they'll have to show that, right? I think their goal, and I don't have the details. It's a great team. I've talked to some of their founding team before. Is to find that virus that would be able to infect or actually work backwards from the indication. Maybe it's a less heterogeneous solid tumor, maybe, but that can infect maybe the majority of cell types. And there is a lot of data to show that CAR T cells are sort of this immune activation. It doesn't necessarily have to be the exact cell it was targeting. So one example, I think TGF beta interferon gamma, which is one of the main outputs of CAR T cells or alpha beta CAR T cells, can actually suppress glioblastoma cell growth. And so it might kill one cell, but some of the sort of spitting out from that locally might actually suppress the growth of the other cells while things kind of do their job.
So Ben, I'm not sure if you remember what I talked about on our CAR T cell versus TCR T cell therapy lecture like a long time ago, but I have an idea that I want to coin the term Gardner T cells. It's a play on Gardner, and I think that we can use CAR T cells. To your point, we wouldn't need to have a CAR T cell that kills every single tumor cell ever. We would just need to become active enough in the tumor to stimulate some kind of broader response inside of the tumor, and you can use them as kind of like gardeners to like lay some fertile soil and not necessarily as warriors to kill everything.
This organoid question is a good one. If I can.
The Remo.
Yeah.
This is actually very interesting.
Oh, well, all of these actually, all of these come into play. I mean, I'm happy to answer that one too.
Let's do Ramos first. Yes.
Yeah. So listen, I think models never answer all of your questions. Never. So for us, in glioblastoma, delivery has already been relatively solved through the intracranial delivery of these CAR T cells. So organoids for us are a way to show that our protection it works. And so you can do this via an organoid model that has more of the tumor microenvironment, more of it represented, not all. Maybe some of them don't have the extracellular matrices, et cetera, but there are other models to answer those questions specifically. So, and importantly, especially for GBM, tumor-driven suppression is the majority of the suppression. But there's a paper that came out just this week that was trying to make that claim. So even if it's a tumor cell line, actually a lot of those suppressive factors are what is being suppressed in the patient. But it is important to have other complementary models to make sure that is the case.
And that would make sense too, because the brain isn't necessarily like a immune hub spot, right? So it's like the tumor wouldn't need to have all kinds of like infiltrative barriers because you're already kind of protected in the brain from the immune system to a certain degree.
Yeah. Super hot topic on immune infiltration into the brain. But that's also a reason I should say that why we're not why we're not worried right now, at least for in vivo approaches, although it's great for other applications. There's not a lot of the infiltration, so T cells really can't even get there, or the delivery can't get there. And if it can get there, it's still being suppressed. So stay tuned for that. It's a really good point, and something that we should be able to have an answer to. But it's important to say models only answer specific questions, and as long as the people know what questions it can and cannot answer, that is one component. And then you might have another model that answers another, and you sort of have to synthesize across the different models.
Right.
Excellent.
Exactly.
Okay. Moving quickly here to Brian's question. What are the newest / best pre-animal models you've seen used to mimic the tumor microenvironment?
Yeah. This is a really, really good one. So I would say organoids have been this maybe widely used verbiage where people haven't had this application yet because it wasn't really ready to go. But one, with the regulatory headwinds recently really wanting to move away from animal models somehow, there's a lot of steam and investment into not only testing drugs via healthy organoid kind of for organs, but also sort of cancerous organoids. I would say that is the best right now that we're seeing are these patient-derived organoids. And there are two versions of this that are super cool. One, you take it from the patient, it's just like broken up and you get whatever you get across 1,000 wells, whatever it might be. There are some companies that are printing organoids. So they can actually take the tumor, somehow separate all the different components and like print really reproducible organoids across each of these wells. And that is a really good representation, but not the best. The tumor microenvironment is not gonna have every component. And if people are working, how do you layer in all these different cell types so you get not only the physical barriers, but all these other suppressive factors we protect from?
Yeah. And that's actually a huge topic in glioblastoma as well. Like doing from imaging, getting like proportions of cells and various things, and like trying to recapitulate intentionally, like painting microenvironments. And even one of the big things with these models as well is we can't properly model the interstitial fluid flow and the pressure and things like that. That's like huge in things like glioblastoma as well. So like you said, no model is perfect. And the more you think about it, the more, you think you got a good model and then it's like, oh, well, we're missing these 14 things. So to try and get to the best one, it's kind of futile, but to get things, like you said, to get data on one thing or a few things from one model and then kind of synthesize a picture from many different metrics, that's the way to go right now.
Yeah, and important to build upon. So everyone has a different story. We are building upon clinical success, maybe small signals of efficacy, but that's how we can work backwards and then use our models to confirm that we've made the right logical kind of decisions, and then we'll learn from that.
Exactly.
So, I think that's why we're really excited to be in GBM, not only to hopefully address this severe unmet need, but it actually is where our science should go first.
Yeah.
Very good. Okay, we've got one more here from Raghuveer. Do these knockout T cells also need immune cell depletion or lymphodepletion before infusion?
Oh, that's a really good question. I think it is to be determined, but I shouldn't say to be determined fully. There has been some early indications that the first versions of these did not lymphodeplete because of this hope that sort of the blood-brain barrier was preventing some of the clearance you might see. It is becoming clear that once these patients develop CAR antibodies that are similar in other applications, that it is really challenging to reinfuse. And so this is the clinical learning what I was saying earlier. That's the clinical team needs to be building upon this to know if that is the case. Do we lymphodeplete, or is there a way to prevent some of those anti-CAR antibodies from coming into play? And also why you shouldn't be using murine-based binders.
Most platforms don't nowadays, though, right?
Correct. Yeah. But it's a really good question. So we don't have plans currently for that, but I also say to be determined because we haven't solidified our clinical strategy, to the T. And I think we are going to we're going to build upon these clinical teams' data where they will be able to inform us because we are not the clinical experts here.
Very good. Okay, I'm not seeing any more questions per se, but some really helpful comments from folks. So thank you for that. One here again that I'll just quickly highlight. FASL has been shown to be at the heart of liver metastasis induced specific CD8 cell death, which is a huge issue for T cell based therapies. So yeah, this is it's getting more and more attention what you guys are working on, Aaron. So this
and maybe the final plug, another thing about METs came up here for delivery. When something metastasizes to another location, its tumor microenvironment usually becomes more suppressive. But that is a challenge, especially if it goes to the brain. So there's a lot of therapies that are starting to work in the breast, lung, melanoma spaces in the primary site. But if it goes to the brain, it then becomes even more suppressive. And that same therapy cannot actually do anything for that secondary site, which is also why we're really excited for this protection approach, where we can hopefully actually go into METs and other indications from backwards into these larger markets by really treating the suppressive environment and broadly applying that backwards. Stay tuned. That's my plug.
Yeah.
Awesome. Well, I think we can wrap it up there. Aaron, thank you so much for coming on. We will definitely have you back at some point in the future. This has been super fascinating. Excited to follow your continued success and all the best. Thanks again for coming on.
And thanks for having me.
Aaron.
Awesome. Aaron, if you can stay on for just a couple minutes once we end the stream, just to make sure everything gets uploaded. Fantastic. All right. Thank you very much, everyone. Have a pleasant afternoon /evening /morning, wherever you are. Bye now.